The most stressed animal in any group is not the one at the bottom. It is the one who knows it. Your brain has been monitoring your social rank every waking moment of your life. Not occasionally, not when threatened, constantly. Right now, as you watch this, a biological system inside your brain is calculating where you stand relative to everyone around you. And whatever it decides is rewriting your brain chemistry in real time. Stanford neuroscientist Robert Sapolsky spent 30 years living with wild baboon troops in Kenya to find out what social rank does to a biological system. What he found is not motivational. It is not something the wellness industry will tell you. It is a clinical fact that explains why your position in any hierarchy is not just a social experience, it is a neurological one. In the Serengeti in the 1970s, Sapolsky began drawing blood from wild baboons. Not zoo animals, not lab specimens, animals living exactly as their biology intended. He measured one thing: cortisol, the primary stress hormone, the molecule the body releases when it perceives threat. What he found restructured the field of stress research. The baboons at the top of the hierarchy had cortisol levels significantly lower than those at the bottom. Not during conflict, not during predator encounters. At rest, baseline. The stress of being low rank was physiologically identical to being under constant threat, even when nothing was threatening them. But here is what made this extraordinary. The high-ranking animals were not just less stressed, they were biologically different. Their immune systems were stronger. Their wounds healed faster. Their telomeres, the biological markers of cellular aging, were measurably longer. Rank was not a social experience. It was a biological one, and it was writing itself directly into the body. Here is what chronic cortisol does to a brain. The hippocampus, the region responsible for memory, learning, and emotional regulation, contains a dense network of cortisol receptors. Under short-term stress, cortisol performs exactly as designed. It sharpens focus, mobilizes energy, prepares the body for action. This is the biological system that kept your ancestors alive. But under chronic exposure, cortisol begins to damage the very receptors it binds to. The hippocampus shrinks. Sapolsky's low-ranking baboons showed measurably smaller hippocampal volumes than dominant animals. Not because they were born different, because their social position produced a cortisol environment that physically altered their brain architecture. Think carefully about what that means. Your rank in any social structure is not just affecting your mood. It is determining the physical size and function of the brain region you use to think, remember, and regulate emotion. The hierarchy is not something you feel. It is something your brain becomes. In the 1960s, British epidemiologist Michael Marmot began studying 10,000 civil servants working in the same buildings, eating similar food, living in the same city. He expected health outcomes to track with income. He found something more precise. Health outcomes tracked with rank, not just income, rank. Each step down in the civil service hierarchy produced a measurable deterioration in cardiovascular health, immune function, and mortality rate. The gradient was smooth and continuous. Every level down, more disease. Every level up, less. What Marmot identified, and what Sapolsky's baboon data confirmed, is that the human brain responds to perceived social position the same way it responds to physical threat. The anterior cingulate cortex, your brain's social monitoring system, cannot distinguish between a real threat to survival and a perceived threat to status. Your nervous system treats being passed over for a promotion the same way it treats a predator. The rank environment you inhabit every day is running a continuous biological program inside your body. Most people never find out it is happening. This is where the modern system becomes deliberate. Social media was built with one specific psychological mechanism in mind: social comparison. The constant, involuntary ranking of your life against the curated highlights of everyone around you. Every metric on every platform, followers, likes, views, comments, shares, is a rank signal. Your anterior cingulate cortex reads each one and calculates your relative standing. A post that performs below your usual level triggers the same cortisol response as a status threat. The platform refreshes every few minutes, giving your social monitoring system a continuous stream of rank data to process. The engineers who designed these systems understood the neuroscience. Former executives from multiple major platforms have confirmed in congressional testimony that engagement mechanics were designed to exploit social comparison circuits. They built a rank environment your brain could not disengage from. Then, they put it in your pocket. Then, they charged you nothing for it, because your attention was worth more than any subscription fee. Now here is what Sapolsky found that changes everything. In the mid-1980s, the most aggressive dominant males in one of the baboon troops he was studying ate contaminated meat from a tourist garbage site. All of them died within weeks. What remained was a troop consisting entirely of subordinate males and females. Animals who had spent their entire lives at the bottom of a brutal hierarchy. Within one generation, the cortisol profiles of the surviving animals changed completely. The chronic stress signature disappeared. The remaining males showed cortisol levels and social behavior more consistent with high-ranking animals, even though the old hierarchy no longer existed. The rank stress had not been an inherent property of those animals. It had been a property of the social environment they were embedded in. Change the environment, change the neurobiology. This is the most important finding in 30 years of field research, and almost nobody knows it exists. There is a second chemical system, rank rewires, one that receives less attention than cortisol, but is equally consequential. Dopamine. The brain's primary motivation molecule does not operate at a fixed baseline. Research by Sapolsky and others documented that social rank directly influences dopamine receptor density in the nucleus accumbens, the brain's reward center. High-ranking animals show higher dopamine baseline activity, more intrinsic motivation, more drive toward goals, more capacity for sustained effort. Low-ranking animals show reduced dopamine receptor density, not because they were born with less motivation, because their social position chronically suppressed a system that requires status security to function optimally. This is the neurological mechanism behind what psychology calls learned helplessness. It is not a psychological response to repeated failure. It is a biological adaptation to a rank environment that signals low status so consistently that the motivation system begins to shut down. The ambition you have lost was not taken by circumstance. It was suppressed by chemistry. Here is what Sapolsky's 30 years of field research ultimately documented. The single greatest predictor of your brain's biological health is not your diet, not your sleep, not your exercise routine, not your genetics. It is your perceived position in the social hierarchies you inhabit. And the modern world has engineered more rank environments than any human nervous system has encountered in its entire evolutionary history. Your workplace rank, your income rank, your social media rank, your neighborhood rank, your body rank, each one running simultaneously, each one feeding continuous data into a biological monitoring system that was designed for one small, tribal group in the African savanna. Your brain was not built for this many hierarchies. The chronic stress you carry is not a personal failure. It is a biological system responding exactly as it was designed, to a world designed to exploit it. Sapolsky documented this in baboons for 30 years before anyone in the modern system wanted to hear it. Now you know why.